Hello, this is Taewoo.kim, technical sales specialist for protein and cell analysis from 써모피셔 scientific solutions. Today, I want to introduce our solutions for 3D imaging and analysis as a title, Total Solutions for 3D Imaging and Analysis. Let's start with 3D research. Sample of 3D imaging, 3D models are very popular these days. 3D papers are still increasing and 3D models are popping everywhere. At first, cancer was the main field for 3D research from 2006 to 2018. However, nowadays, researchers using cell sources from not only major organs but up to kidney and heart, though very small portions. It means we can make 3D models almost every organ. There are two major types in 3D models. One is spheroid and one is organoid. Big difference is cell source. Spheroid is mainly using immortalized cell line and just 3D cell aggregate. Otherwise, organoid is made from stem cells, iPSC, other temporal cells. Therefore, organoid can differentiate and got functionality to mimic human organs. 써모피셔 can provide almost every sources for 3D research. We have cells, special gel matrices, and cell culture plates including U-bottom, Nunk brand, and very well-known media and supplements too. In addition, we have instruments and reagents for 3D imaging and analysis. Today, I will mainly focus this area. Let's talk about 3D imaging and analysis. Do you have any experience for 3D imaging and analysis? I think most people attended this seminar have an interest to 3D imaging and analysis. How do you think about 3D imaging and analysis? Is it difficult? Because 3D structure is very thick and not easy to imaging? Or easy like just 2D imaging? I want to say not difficult. I cannot say easy just 2D imaging. However, not difficult if you have right platform. Therefore, I want to introduce our platform. This is brief introduction of 써모피셔 scientific solutions. We are the world leader in serving science. We have very well-known 6 brands, Thermo Scientific and Patheon. We can provide a lot of instruments and reagents and including pharma services now. I want to more focus imaging. 써모피셔 is an expert of imaging. As you can see, molecular probes, which developed very well-known fluorescence dye in 1975, is in part of 써모피셔, and we merged AMG and Cellomics to develop EVOS and HCS. Therefore, we are the only one can provide both reagent and instrument for imaging. This is the slide that 써모피셔 is the first inventor of high-content screening platform. As you can see, the company Cellomics developed original concept for high-content screening at 1996, and this Cellomics is merged to 써모피셔 Scientific at 2006. After 2006, we developed and launched many new platforms up to CX7 laser. Today, I will focus these two platforms, high-content screening, CX7 laser, and reagents including CytoVista, Alexa Pro Plus, Cell Event, LiveDate, and CellLogs. First, I will introduce high-content screening. This is kind of definition. High content means a platform for understanding of phenotype changes of population of cells through microscopic imaging and analysis. High content technology provides simplified workflow for cell biology and robust data handling. Therefore, researchers can easily understand complex phenotype of cells. This is comparable high content screening to conventional cell imaging. High content screening is based on cell phenotype, though using same plate. The result from high throughput screening is simply kind of binary type. However, as you can see in the right panel, high content screening can provide complex throughput analysis data. In addition, we can provide automated imaging, analysis including quantification, data management, and data organization and visualization. Here's a lineup of our high content screening instrument. We have 3 models, CX5, which has 5 LED light sources, kind of entry model of high content screening platform. CX7, which has 7 LED wall cell laser and confocal is integrated. Therefore, this is for emerging assays like 3D. CX5 and CX7 have same analysis HCS software and high resolution camera and store data management platform. In this talk, I will focus on CX7 laser. This is specification of CX7 laser. Maximum magnification is 60x. It means 600x magnification. We have on-stage incubator for live cell imaging, confocal color module for imaging of color sample, and laser autofocus, high resolution camera, and 7 lasers. From next slide, I will focus more in detail. Okay, first laser. We have 7 lasers for excitation. Like left pictures, inside of CX7 laser, there are 7 individual lasers with fibers. Therefore, we can diminish loss of power. And you don't need any kind of pre-warm-up, and you can control laser power with software. And in addition, we are the only one which provide 7 lasers in high-content screening platform. This is advantage of laser compared to LED. Laser excitation is more powerful and narrowband. Therefore, laser excitation can provide more specific and clear fluorescence image. This is our laser and fluorescence spectrum. We have specific 7 lasers from DAPI to neo-IR, and we have multiple color filters. Therefore, we can provide blue, cyan, green, yellow, orange, red, far red, and neo-IR. This is example of laser illumination. Same sample using LED and laser. Compared to LED, using laser illumination, you can see more clear in detail of cell structures like narrow fiber type sample. CX7 laser has confocal inside. We have spinning disk confocal, which is known that good for biosample imaging, and we provide dual pinhole 70 micrometer. You can choose pinhole up to your sample's type, and major advantage is customer can mix wide field and confocal imaging according to each fluorescence channel. For example, you can use wide field for DAPI and confocal for only GFP. Therefore, you can save time and laser consuming. This is result of confocal at just 2D imaging. Confocal can make image fluorescence more specific and clear in detail like spot and even low magnification. This is just from 20x magnification. And you can use G sections and make projection images. We provide several types of image projection, but mainly use maximum projection. Therefore, using confocal and G section, you can make clear 3D imaging like this. Here is a summary of laser illumination. Laser allows more light to sample, therefore more feasible for 3D imaging. Laser illumination can provide best performance. According to previous slides, laser illumination increases imaging speed, image quality at wide field and confocal. Therefore, we launched 7 laser integrated CX7 laser platform. I think the analysis software is the core of high-content screening platform. HCS Studio is our analysis software, and here is application what you can do. As you can see, there are already more than 30 applications already set up and validated by references. All application is iconized and just click what you want to do. In addition, you can set up your own protocol. Last 2 slides is huge advantage of our high-content screening platform competitors. First, I can guarantee we can provide the first imaging and data faster than any other competitors because we can do imaging and analysis simultaneously. Sequential process imaging first and analysis next is very general process and everybody doing. However, we can provide simultaneous imaging and analysis. Therefore, customer can receive the first result right after imaging. The last one is customer can receive imaging data at single cell level. In this figure, all green dots in left field are results from cells, in a well, for example, and all imaging results are linked to own cells. Therefore, you can validate result each cell level one by one. So we can say each cell has own story. You can use this function to validate your data. For example, where comes this data? In addition, you can gating cells according to specific condition. We can provide this function no matter how many wells you used. Through our platform, you can receive important parameters for 3D imaging. You can do 3D imaging and analysis using colony formation assay or shape and phenotype analysis. and spot detection for inner analysis for 3D structure. First 3D parameter is area. You can receive area of 3D spheroid or organoid. Second one is shape analysis. We have three parameters for shape and volume. P2A, ratio of parameter and area of the object. LWR, length and width ratio of object. In these two parameters, one means very round shape, and numbers going far from 1, it means shape is going to non-round. Last one is volume of 3D sample. This is the result of P2A. You can divide round and non-round shape according to P2A ratio. And this is the result of LWR. You can distinguish, for example, oval shape and round spheres. As you can expect, this is the data of volume. You can check a spheroid or organoid level. You can receive not only bright field, you can do analysis inside of 3D spheroid and organoid. This is the result of dead cells imaging in multiple organoid sample. Okay, let's move to second platform, reagent for 3D imaging and analysis. If you are doing 3D fluorescence imaging, you will definitely encounter a situation like this image. It is very difficult to see fluorescent signals from deep area of sample because 3D models, spheroid or organoid is very thick and opaque because there are many cells and tissues are located inside of sample. Then, how to overcome? The answer is clearing. Clearing means that you make sample transparent to allow the light can penetrate into a sample. We have a clearing solution named CytoVista. Refractive index of CytoVista is adjusted to protein or tissue. Therefore, you can receive clear fluorescence image. Then how can you use? It is very easy. Just put sample into the solution. Maximum thickness for clearing is 10 mm and clearing time is up to size. For example, this kind of red brain it needed 48 hours. After clearing, you can see labeled cells over 400 micrometer. After clearing, you can get more clear image at deep area of 3D sample and receive more clear numbers all part of 3D spheroid. You can see differences of counted cells number between cleared spheroid and not cleared spheroid. This is specification of CytoVista. You don't need any special instrument for clearing and it is to use. Just follow protocol in the manual and no repeated extraction. No repeated extraction can maintain sample shape and rigidness. The right picture of this slide is 4 types of clearing method. Each method has their own advantages and disadvantages. CytoVista is like B, simple immersion. You can see there is no repeat extraction compared to other models. Here is example movie using CytoVista. You can get this kind of clear image for your 3D sample or thick tissue. Left one is 3D rendering of 1 mm mouse prostate tissue section, and right one is 500 micrometer diameter StemoniX 3D microbrain organoid, which generate from iPSC-derived neural progenitor cells. Second reagent is Alexa Flow Plus. Alexa Flow is the famous and powerful flow probe because brightness is very strong and photostability is also good. And there are multicolor flow probe from DAPI to Neo-IR as like right figure and very stable and easy to use. Therefore, Alexa Flow is the gold standard for fluorescence imaging. In this year, we launched Alexa Flow Plus. Alexa Flow Plus provides up to 4.2-fold higher signal-to-noise than Alexa Flow. And here is an example of Alexa Flow Plus. 408 green, 594 red, and 647 far red image. I think Alexa Flow Plus is good for 3D imaging because 3D sample is more thick and opaque than 2D cells. I want to finish this talk introducing our case studies using our platform. Okay. First one is imaging and analysis of necrotic core of spheroid. We use live-dead reagent for labeling dead cells of Hep-G2 spheroid, and we use CytoVista for clearing solution. As expected, we can counted green dead cells as increasing seeding cell numbers of spheroid, and using our CXM platform, we measured distance from center to dead cells or DAPI cells. You can see ratio of dead cells to DAPI is close up to 1 as increasing cell numbers as matched to images. Second one is spheroid phenotyping measurement using cell event reagent. Cell event can label apoptotic dead cells in 3D spheroid. According to increase of niclosamide dose, caspase activation is increased while spheroid size is decreasing. The third one is detection of RS level in spheroid. We use Cellox DIMRAD. This DIMRAD is very sensitive to RS level and very stable to maintain fluorescence more than any previous RS detection dye. We can measure the RS level fluorescence signals after treating Menadione and measures this fluorescence intensity according to Menadione dose. Okay, I want to wrap up this talk with this slide again. I introduced our platform, high-content screening CXM laser, and reagent you can use for 3D imaging analysis. I think you can easily start an approach to 3D imaging analysis using our platform. Thank you for attention today. If you have any comments or questions, please send me email. And thank you.